So my name is Emily Fields. I'm going to talk to you all this afternoon about some of the research I did as an undergrad student at Guelph. So I looked at species differences in response to light and their implications for selective passage of desirable fishes at in-stream barriers. So the connectivity conundrum is at the core of the study and at the field of selective fish passage as well. So it's important to know what it is in order to fully understand the goal of this study. Currently, 60% of rivers worldwide contain at least one in-stream barrier. On one hand, these barriers prevent native species from reaching foraging, spawning, refuging habitats. They reduce their overall fitness, their productivity, and the biodiversity. Alternatively, these barriers also prevent further invasion from invasive species and reduce diseases or contaminants from further spreading within river systems. So the connectivity conundrum arises when the best management action for maintaining biodiversity and production and facilitating or restricting connectivity between river sections is unclear. Selective fish passage of desirable fishes and blocking of invasive fishes at in-stream barriers could resolve the connectivity conundrum, but it requires efficient mechanisms for sorting desirables from invasives. So while current selective fish passage technologies are successful in solving this issue, there are inefficiencies that can be addressed and improved by integrating multiple sorting mechanisms. So currently the literature suggests that we could achieve greater sorting efficiency by developing multi-factor fishway systems that combine multiple sorting processes targeting different traits, which would allow river managers to create filters that promote the passage of one species over another. Additionally, previous studies that have assessed sorting mechanisms have aggregated the response outcomes of the desirable species, ignoring species-specific responses to treatment. So the results of this study will help provide stakeholders with additional methods to improve current selective fish passage technologies. So this past summer, we tested experimentally if desirable fishes differed in their responses to light in a two-chambered trap and sort fishway that was used to sort invasive sea lamprey from desirable fishes at an inflatable sea lamprey barrier on Big Carp River near Sault Ste. Marie. A stratified random field experiment was used to test if the manipulation of light within the fishway could help improve rates of sorting efficiency. So this is kind of a brief overview of the experimental design within the fishway. So the study period of trap operation was divided into 18 two-night strata. Within each stratum, a lit treatment was assigned randomly to one night and an unlit control treatment was assigned to the other night. So the lit treatment consisted of lighting the downstream chamber using a modified LED mechanics light and then adding a light refuge at the bottom of the upstream chamber. So the research question that we investigated was, is including taxonomic affiliation important when determining sorting efficiency rates within fishways? So we hypothesized that including taxonomic affiliation within the model will allow for more accurate estimates of sorting efficiency by examining differential responses in sorting efficiency among these desirable fishes. So we investigated our hypothesis using 3 models, which are what you see at the top of your screen. So model 1 didn't consider taxonomic affiliation and just assumed that there was no differences in sorting efficiency among all desirable fish. Model 2 considered taxonomic affiliation as a random intercept to examine baseline differences in sorting efficiency. So this assumed that species differed in sorting, but did not in response to treatment. And then model 3 considered taxonomic affiliation as a random slope with respect to light treatment to test for differential responses to light across species. This model assumed that species differ both in their sorting and in their response to light. So we took a kike information criterion values from 3 generalized linear mixed models and compared them to find the best approximating model. So the results from this analysis revealed that model 3, which was the random slope model, was the best performing model in the model set. As this model carries 94% of the cumulative model weight, it was very likely that this model provides the closest approximation of the truth of what's occurring in our data. So as such, we accepted our hypothesis that taxonomic affiliation does increase predictive power of the sorting efficiency model and does allow for more accurate estimates of sorting efficiency. So model comparisons based on a Kike's information criterion demonstrated that desirable fishes differed in their response to light. So if you look at these graphs here on the y-axis, we have sorting efficiency, which ranges between 0 and 1, with 1 being 100% of fish successfully passing the barrier. So if you look at the random slope model on the right-hand side of your screen, we can see that some species experienced increases in passive sorting while others experienced overall decreases in sorting. Specifically, we saw very extreme reactions in response to the light from Johnny Darter and mottled sculpins. So Johnny Darters experienced a 42% increase in sorting efficiency under the light treatment, while mottled sculpins experienced a 38% increase. So there were several possible explanations as to why we saw these two species experiencing such strong reactions. Diurnal fish species, such as the Johnny Darter, have phototopic vision and are generally inactive at night due to a perceived threat of predation. But if the fishway was lit using artificial light bars, then this species may have been more likely to attempt passage due to a decreased predation threat. Additionally, several studies have noted phototactic responses for mottled sculpin, indicating that these species were more attracted to the downstream chamber on nights when light treatment occurred. So in conclusion, the study confirms that taxonomic affiliation is a very important parameter when assessing fishway efficiency and rates of sorting efficiency among desirable fishes. Including taxonomic affiliation allows for the improvement of these models as it accounts for differential responses among desirable species. These results indicate that we are seeing species-specific responses to light treatment and are observing differences in both the magnitude and direction of response. So this implies that there is some degree of self-sorting occurring within the fishway in response to light. Several benthic species were observed to have strong responses to light treatment, indicating the potential presence of a relationship between these individuals' behavioral traits and the use of artificial light. So in terms of what's next, this study provides evidence that the use of light as a filter in selective fish passage is continuing to be a very promising area of research and will aid in the development of future passageways. Understanding how fish behavior changes with light intensity will help river managers and other stakeholders make more informed decisions on in-stream barrier design. And future fish passageways can be designed to target the relationship between benthic species and altered light as a new invasive species management strategy. For example, the round goby and mottled sculpin share similar foraging behavior, habitat use, and growth morphology. So using a light treatment system in the fishway could be beneficial in increasing the passive sorting of these invasives from desirable fishes. So that concludes the end of my presentation. I'd like to say thank you to my supervisors and the field staff who assisted on this study, and thank you all for listening.